Welcome to Microscope chapter part 1, Optical Microscope. In this chapter, we will cover the basic concept of microscopy and discuss various types of microscope including simple microscope, compound microscope, dark-field microscope, phase contrast microscope, and fluorescent microscope, and also explain their fundamental working principle and functions. Microscope. Microscope is an optical instrument consist of lens or combination of lenses for enlarged or magnified image of minute objects. Microbes are too small to see through our naked eye, so with the help of microscope, we magnify the image and observe. Because our resolution power of our eye is only 200 micron, and the size of medically important bacteria is 0.2 to 1.5 micron in diameter and 3 to 5 micron in length. So, with the resolution power of our eyes, it is difficult or not possible to see the microorganism. So, with the microscope, we stained or enlarged the magnification of the object or microorganism. We observe and we can study the morphological characters of a microorganism with this help of this microscope. Resolution power. Resolution power is the ability of an instrument or our eyes to distinguish the object that are closer together. Here you can see these 2 points we cannot observe through our naked eyes, but when we go here, these 2 points can be resolved. Okay. It can refer to the ability of an optical system to form separate image of object. They are closer together. So this is called as resolution power. And next about the size calculation, micron, nanometer, angstrom. Micron is equal to 1000 of millimeter. Nanometer is equal to 1000 of micron, angstrom equal to 1/10 of nanometer. This is the scale. Here you can see 1 centimeter. 1 centimeter is equal to 10 millimeter. So then let's come to the micron. The 1 micron we already told that it is 1000 of millimeter. So 1 micron is equal to 1000 of millimeter. That means we will take 1 millimeter and we will equally divide this 1 millimeter into 1000 times. From that one part we will take, that is will be equal to 1 micron. And mainly the medically important bacteria is measured in micron or micrometer. Next about the nanometer is equal to 1000 of micron. That means we will take 1 micron and we will equally divide this 1 micron into 1000 times. From that one part we are taking, that will be equal to nanometer. 1 nanometer. And most of the virus, not only the most of the virus, all the virus is measured in nanometer. At last, the angstrom. Angstrom is equal to 1/10 of nanometer. That means we will take 1 nanometer and we will equally divide this 1 nanometer into 10 times, and one part we are taking, that will be equal to 1 angstrom. The angstrom are measured in atoms. Microscope. The microscope is divided into simple, compound, dark field, phase contrast, fluorescent, and electron microscope. And except this electron microscope, all are optical microscope, and they are illuminated with the help of light rays. But in the case of electron microscope, electron beams are used instead of these light rays. First, about the simple microscope. Simple microscope, that means in the name itself, it is clear. It is very simple. A simple microscope has only one type of lens. Here you can see this is the lens. Through the lens, we will observe the sample or specimen, and this is the sample keeping area, or we can say this is stage, and this is the light source. Light source here used as mirror. Mirror is used as light source. So when light will come and hit the mirror, it reflects the light. So the light will reaches here, the stage where the sample is kept. And through this lens, we can observe the minute particles or minute object here. And in the simple microscope, we can examine minute algae, fungi, and biological material. Compound microscope. The compound microscope is not a simple microscope, because here in compound microscope, combination of lens are used, and ocular and objective lens are used. And this objective and ocular lens will magnify the image, and we can observe or see the enlarged image with clear pitch. And these are the parts of compound microscope: ocular lens, body tube, arm of the microscope, nose piece. The nose piece, you can see the objectives. This is the stage, and this is the clip. This is the condenser, diaphragm, light source, coarse adjustment, fine adjustment, and base. And these are the parts of compound microscope. We'll go in detail about that. This is body tube, and this is the body tube. The body tube holds the objective lens and the ocular lens at proper distance. And here is the nose piece. The nose piece holds the objective lens and can be turned into increase the magnification. And this objective lens is connected in the nose piece. By rotating the nose piece, we can select the different magnification of objective lens. And this is the ocular lens. And through ocular lens, we can see the enlarged view of the object or the sample. And this is the arm of microscope. It is used to support the microscope when carried, and also hold the body tube, nose piece, and objective lens. Objective lens. The objective lens increase magnification of the image. In microscope, there are two types of lens are used. First one is objective lens, and this is the ocular lens or eyepiece. So when the light rays passes through the specimen, it will enters into the objective lens. So the objective lens will magnify the image. In compound microscope, we used different type of objective lens depending upon the magnification what we need. And in microscope here we use 4x, 10x, 20x, 40x, and 100x. And this objective lens is present in the nose piece. And by rotating the nose piece, we can change the magnification of objective lens. And this is the stage. The stage will support the slide, and here in the stage we keep the specimen. Stage clip. These are two stage clips hold the slide or specimen in place on the stage, and simply the stage clip will hold the slide. The condenser. The condenser focuses the light and illuminate the sample. This is the condenser. When light passes through the condenser, condenser will focus the light into the sample. Also, it will illuminate the sample. So we will get a clear or better image. Diaphragm. This is the diaphragm. The diaphragm controls the amount of light on the slide or specimen, or also we can say that the diaphragm will control the intensity of light to the specimen. Coarse adjustment. Move the body tube up and down for focusing the image. So that means the coarse adjustment will help us to focus the image. Fine adjustment. This is the fine adjustment. This knob moves the body tube slightly to sharpen the image. That means first, with the help of coarse adjustment, we will focus the image, and with the fine adjustment, we will sharpen the image for clear visibility. Light source. Project light upward through the diaphragm, the specimen, and the lens. And this mirror an electric bulb are used as light source in earlier times, or in the basic simple microscope we used mirror as light source. So the light will come and hit this mirror. The mirror will reflect the light towards the specimen through condenser. And nowadays in most of the microscope we used electric bulb as light source. At last, this is the base which support the microscope. So the principle of compound microscope, and these are the parts of compound microscope. This is the light source, and this is the condenser. In between condenser and light source, there is a diaphragm is there, and this is the specimen stage. We will keep this microscope stage. Here we will keep the specimen, and these are the objective lens, and these are the ocular lens. So first, the light will passes through the diaphragm. The diaphragm will control the light, or a diaphragm is having a shutter. If you close the shutter, the light will not passes through this diaphragm. It will not reaches to the condenser. So by adjusting the shutter of the diaphragm, we can control the light. So the light will reach us to the condenser. So what will happen? This condenser will focus the light to the specimen, and also it will illuminate the specimen or the object. Okay, and after that the light will reach us here, the objective lens. So the objective lens will magnify the image. There are different type of objective lens are there. We already discussed earlier, 40x, 4x, 10x and 100x. Depending upon the need, we can change the power of this objective lens. Or simply we can say that this objective lens will multiply or magnify the image. When this light rays passes here, reaches the ocular lens, or this ocular lens will also magnify the image. And from here, with the help of our eyes, we can see the enlarged view of the microorganism or the object. Magnification. Magnification is also called as the power of microscope, or enlargement of microscope. We will call it as a magnification. In microscope, we already told that the tiny object or microorganism is magnified for better observation. In compound microscope, there are two types of lens are used. One is this is the objective lens and the ocular lens, or we can say that this ocular and this objective lens will enhance or magnify the image. And the total magnification of the microscope can be calculated by multiplying the power of ocular lens and objective lens. And in compound microscope, there are single type of ocular lenses there, mainly that is in 10x, and different type of objective lens are used, that is 4x, 10x, 40x and 100x. And this lens can be changed depending upon the need of our observation. So in magnification power of low power is 100x. In low power, we can observe the protozoa and larger microorganism. And this magnification power of high power is 400x. With the help of this high power, we can observe the motility of microorganism, mainly motility of bacteria, protozoa and fungi. In oil immersion, the maximum magnification is 1000x. With the help of this oil immersion microscope, we can stain the smeared, or we can observe the stained smear. Oil immersion. In oil immersion of compound microscope will give high magnification power up to 1000x. Oil immersion we use to observe stained bacterial smear. Not only the bacterial smear, stained smear we can observe in oil immersion microscope or oil immersion objective. This will give a clear image. In oil immersion, we place an oil between the smear and this objective lens. By that, we will get a clear image of the organism. We can study the morphology of stained smear very clearly, because oil will fill the gap between the smear and objective lens. Because of the same refractive index of oil will avoid the refraction of light. That is, all the light will passes through the smear will reaches the objective lens. So we will get a clear image of the magnified picture. So this is the oil immersion light. This is the light source. The light source will pass us through this oil. While passing, what will happen? The scattering of the light or the refraction of light will be limited or controlled. So all the light will reaches to the objective. So we will get a better image. And here you can see, due to the absence of this oil, the light will be refracted or scattered, and only some amount of light will reaches to the objective lens. Darkfield microscope. In this type of darkfield microscope, a special type of condenser is used with a central circular stop. So this condenser will illuminate the object, and when the light reaches here in ocular lens, we will get a clear image of the object with a dark background. In darkfield microscopy, a special type of darkfield condenser is used with a central circular stop. This condenser will illuminate the object. That means the light source will pass through this opaque light stop to reach us to the condenser lens. This condenser lens will focus the light, or it will illuminate the sample. When the light passes through the sample, what will happen? There will be a scattering of light will be occur. The scattered light will reaches to this objective lens. So this objective lens, again it will illuminate the sample or the organism against a dark background. So we will get a clear, visible microorganism with dark background image. And this darkfield microscopy is used to demonstrate slender organisms like spirochetes, Vibrio and Campylobacter in specimen and in culture, microfilaria in blood. An ordinary microscope can be converted into darkfield microscopy by using this special type of darkfield condenser and a central circular stopper and a funnel stop for insertion of this 100x objective, so that the simple or the compound microscope can be converted into darkfield microscopy by adding this special type of condenser. Phase contrast microscope. In this microscope, improve the contrast and make evident the structure within the cells due to the difference in thickness or refractive index of the specimen. The difference in refractive index between the bacterial cell and the surrounding media makes them clearly visible object. In phase contrast microscope, we use a special condenser with annulus and a special phase objective with a phase plate are required for this difference in the phase. When light rays are passed through an object, they emerge in different phase depending upon the difference in the refractive index between the object and the surrounding medium. These phase differences are converted into difference in intensity of light, producing light and dark contrast in the image. And this is the principle of phase contrast microscope. This is the light source. The light source will pass us through this annular ring, or then the condenser ring. This is the annular ring. The condenser will illuminate or focus the light when the light reaches to this specimen, there will be a refraction or scattering of light will be occur depending upon the intensity, or we can say depending upon the thickness of the specimen. When the thickness of the specimen is different, the passing of light or the scattering or the refraction of light will be different. So the scattered or refracted light will reaches to this lens or the objective lens. The objective lens is having this face plate. This face plate will segregate the direct and scattering light. So through this ocular lens, we will get a clear, enlarged image. In this phase contrast microscope, when light passes through cell, small phase shift will occur, which are invisible to our human eyes. In a phase contrast microscope, these phase shifts are converted into changes in amplitude, which can be observed as difference in image contrast. So by the help of this phase contrast microscope, we can observe unstained living organism with good contrast and high resolution. And also it will be useful for the study of structure and also the structural changes in larger microorganism and tissue cell, and also the slender object like spirochetes and all. So by the help of this phase contrast microscope, we can observe live or unstained microorganisms clearly due to the difference in this phase. Fluorescent microscope. When fluorescent dye are exposed to ultraviolet rays, they produce fluorescence. And fluorescent substance absorb UV light and emit visible light. And also we can say that they convert the invisible short wavelength rays into light of long wavelength. And this is the basic principle of fluorescent microscopy. Mainly we can say that in fluorescent microscopy, we used fluorescent dye to coat the specimen. So when fluorescent dye are exposed to an ultraviolet rays, they become fluorescent. And that fluorescence we will observe to the ocular lens. The working principle of fluorescent microscopy. And here this is a specimen. This specimen is already coated with this fluorochrome or fluorescent dye. When a UV light is exposed to this specimen, what will happen? The specimen will start illuminate. The fluorescence will be produced and it will emit the fluorescent. This emission of the fluorescence can be detected in the ocular lens. So we will get an image like this. In a dark background, the fluorescent coated bacteria or the specimen can be seen like this. And this is the picture of spirochetes, the fluorescent coated spirochetes in dark background.
Subscribe to Simply Micro Med. If you're curious about medical microbiology, infectious diseases, and unraveling the mysteries of tiny organisms, hit that subscribe button. Join our community of science enthusiasts, and let's explore the microscopic world together. Remember, knowledge is contagious. Spread it by liking and subscribing. Thank you.